address each one of you by his first name and title, but I do not know all of you, and therefore I will start with an introduction saying, dear members of the Homo sapiens, welcome to this fantastic environment, and I'd like to thank very much everybody that gave me the honor of inviting me, but particularly Professor Dr. Ioannis Patrikios and the Scientific Program Committee for the kind invitation. I also want to thank the members of our laboratory and the funding agencies that supported our research over the years. The Swiss National Science Foundation, the European Union, a specific institution within the European Union called the ERC, the American NIH, and 5 private foundations, Ludwig, Gebert, Lejeune, Provisu, and Childcare, for the support over the years. When I go to dinners, like all of you, people ask me, what do you do in life? And I usually say, I'm a geneticist. And they say, what is this? And I say, this is a matchmaker. A matchmaker that matches functional genomic elements in the genome, genes and other things that we have in the genome, and phenotypes, disorders. You can see them as squares on your left and circles on your right. And what a geneticist usually does is links these two objects, the genes in the genome and the disorders. And the arrows that link those are the mutations, the variants that we all have in our genome. And I will talk to you a little bit more about this. This game, the matchmaking game, is, as my daughter told me a long time ago, that's very simple. Well, it is very simple when one gene relates to one disorder. But most of the times, this is very complicated because one gene relates to many disorders sometimes, and several disorders relate, the same disorder relates to many genes very often too. Now, what is the genome? The genome is all the DNA that all of us have in each one of the trillion cells in our body. And the 1st of October of 1990, a long time ago, there was a project that started that we used to call the Human Genome Project. It started that day because at that day, the American NIH put $120 million for that project. And the goals of this was to map the genome, to clone the genome, and to sequence, to read the genome of humans. That took 12 years to do all this international project. And in the middle of 2002, there was a big feast at the White House. And I remember at the time was President Bill Clinton that you see in your screens. And the Prime Minister of the United Kingdom on a big screen like this. It was the first time that I saw a teleconference of Tony Blair on the screen. And at that year, we celebrated the reading of the human genome. Of course, everybody knew that we haven't finished with the genome yet. But nevertheless, that year, that day was a big achievement because since then, we live in the post-genome era of medicine. So the genome is a tremendously big molecule. Let's compare it to a text in a book written by 4 letters, A, G, C, and T. 4 chemical substances, one after the other, and they make the genome. And the genome is, do you want the number? 6 billion letters in each one of the trillion cells on our body. It's enormous. However, it is finite. So in other words, there's a beginning and an end. And if something is finite, that's very scientific. We can measure it, we can read it. Not us with our eyes and brains, but the computers can do this. And therefore, this is something that we can study and learn from it. So here you see a small part of my genome. It's 10,000 letters. The genome that of you and I, as we said, is 6 billion letters, is about 300,000 times bigger than this. So this is that molecule that changes medicine every year. Of course, as I said, at that day, the 2002, that was not the end of the sequencing of the genome, because we did not know the most difficult regions. That was about 8% of it. And it took us another 20 years to finally sequence the entire genome. 1st of April of 2022, using new technologies and advances in computing, we have the final reading of our genome as humans. And for the aficionados, for those of you who know a little bit more about chromosomes and cells and genomes, those difficult regions were the short arm of human acrocentric chromosomes, 13, 14, 15, 21, and 22. A big region on chromosome 1, big region on chromosome 9, chromosome 16, plus the human Y chromosome that was almost unreadable in the beginning. So now we're done with this. And that added several genes and several functional genomic elements that we will study in the next 30, 40, 50 years. Now, 6 billion letters is a big molecule. And some people ask me, can we have an example of how big is that genome? Because when we say millions or billions, etc., sometimes it's beyond our grasp. So the genome in our cells, it is divided into chromosomes. The chromosomes are, think of the chromosomes as the buses that transport the genome from one generation to another or from one cell to another. And the smaller bus, a mini bus, is the smaller chromosome. That's called chromosome 21, has a number. Chromosome 21 is my beloved chromosome. My laboratory studies this for a long time. And it's about 1.5% of the genome, 1.5%. So I convinced the Swiss Institute of Bioinformatics to print me the genome, the DNA of chromosome 21, which is 1.6% of the genome. And they produced that book that has 14, 17 pages and then about 30,000 letters per page. The letters, the A, G, C, and T that you will see, there are size of 7 when you type your texts, usually type 11 or 12. That's one copy of this book, and I have it with me, and I pass it to you to take a look. So here it is. It is about 4 kilograms. It had cost me a lot of money with the luggages over the planes, but it's worth it. So here is part of it. You can see all the text. So I give it to you. Each one of your cells, of our cells, of the trillion cells that we have, has a text that is 66 volumes of this. 66. I start with you. Thank you. So that's an enormous molecule, and then I come back to the work or the job of a geneticist, the matchmaker, And I don't know how many of you have seen that movie, The Fiddler on the Roof, 1951, from Norman Jewison, who died like about a year ago. Can we play the movie, please?
Matchmaker, matchmaker, make me a match.
The lady
Find me a find,
like
catch me a catch. Matchmaker, matchmaker, look through your book
Look through your book.
and make me a perfect match.
So the book is the book of DNA that you have over there. And the object that we match is what is important in our DNA and how that translates to phenotypes and disorders. So here is a couple. And you see on blue the DNA molecule of one partner, the blue partner, and the DNA molecule of the pink partner. And every time we make the genetic experiment to have a child, we shake the DNAs of the two and we take half from the father and half of the mother, we mix them up, and we have a new individual. However, in order to do this, we need to copy the DNA. We need to copy the DNA in the cells in order to give it to the next generation, or in order to pass it from one cell to another. And the copying of the DNA is extremely accurate. It is extremely accurate. If it was not accurate enough, we wouldn't have children that look like humans. But when you see a pregnant woman in the streets, you're thinking that this woman will have a child, not a cat or a cow or a Drosophila or a fish, but a human in her womb. Why is that? Because she and her partner gave that new individual half of the DNA that is human. The copying is quite accurate. I'm talking to you now for about 5-6 minutes, and millions of my cells in my gut and the blood have been renewed. So that copying mechanism worked on me continuously. And a cell takes about 20 minutes. Think of this, 20 minutes to read and copying correctly 6 billion nucleotides. The copying is very accurate. However, there are mistakes that are made. The mistakes are very few. In our language, we say 10 to the minus 8 per nucleotide per generation. For you is one letter change, mistake, in 100 million letters. So that's the error rate. So when I was born, I had the DNA of the zygote. No, when I was conceived, I had the DNA of the zygote, and then every time there was new cell made on me, then I added mistakes. And you see the mistakes in the scheme there. Every time you copy the DNA, you add few of the circles that are red, so you accumulate variants. In me, when I accumulate variants, I age, and my cells differentiate a little bit. In humanity, that means that every child has new mutations, new variants. And then we have the evolution of humans as we go through our history. And that has as a result that the DNA of you and I is not the same. We're all part of the Homo sapiens sapiens. So we have the human DNA. However, we are very different because our DNA is a bit different. So here, you see with the little flags, the letters that are different in my DNA from the genome of reference that we call it. 1 in 1,000 letters varies between two randomly chosen genomes in the population. And that contributes to the differences that we all have. There are not two people on Earth, among the 8 billion people that we are on Earth, that have identical genomes, except monozygotic twins when they are conceived. Even the monozygotic twins change during their life because they accumulate somatic variants. So we all humans, because we share our genome, but we have a difference. 0.59% is the average difference between me and anybody else on Earth, or you and anybody else on Earth. This is an important difference, though. It is an important difference because that makes us all different, with different abilities and different facial and somatic characteristics, different orientations, different likings, voting difference, etc., behavioral difference. And that is fantastic. That's a gift of nature. It is a gift of nature that we're not cloned or clones of each other. We are very different, and that is a great gift of nature. It is a great gift because this genomic variability provides the possibility to evolve and to adapt. Because the environment changes, and we need to change in order to adapt. And biological species change, and we also need, as humans, to evolve in order to survive in the ever-changing environment. However, gifts, they don't usually come as gifts. There's no free lunch, as we say. And as humans, as every other species on Earth, we pay a price for this possibility to evolve and adapt. And the price that we pay is that sometimes this variability is bad. It's not good for us. It's bad. It's pathogenic. And it's pathogenic, and it gives us the myriads of human disorders that we call genetic disorders. So the genomic disorders is the price to pay for the potential of evolution or adaptation. This big and obvious secret in medicine is that the genomic variability, along the environmental variability that we talk a bit later, is at the root of every human trait and phenotype and disorder. And as this very big and famous population geneticist, Theodosius Dobzhansky, wrote in a book in 1973, nothing in biology makes sense except in the light of evolution. And us, the medics, we can paraphrase this and say nothing in medicine makes sense except in the light of evolution. So here are my variants, or anybody's variants, and the variants that we have in our genome, the mutations, come in 3 flavors. Some variants are very rare. We call them the pathogenic, high impact, because they produce severe genetic disorders. Other variants, they're common, but they're not innocent. They predispose us to the myriad of adult onset disorders, predispose us either positively or negatively. In other words, some of them, they protect us. And we also have another big category that we call neutral. These are fossils of the evolution of us. And the big question in the next 50 years, it will be which variants are predisposing, important, and which variants are neutral. Genetics is, at the end, very serious business. And I'll show you an example of this. Could you please play again? So here's an athlete. He looks healthy, contributing to the game. But all of a sudden, it drops dead. It drops dead because it has a rare genetic disorder that's called hypertrophic cardiomyopathy, which is, in his case, due to one letter difference in its DNA. Can you think of this? 66 books like the ones that I showed you, with all the letters, and one of them is different, and that one of them gives this individual and his family this very serious phenotype that we call hypertrophic cardiomyopathy. And this is one of the hundreds and thousands of disorders that we call Mendelian genetic disorders, and the hundreds and thousands of other phenotypes and disorders that we call complex common phenotypes of later age. I'll give you an example of the genetic determinism. The variants can be very deterministic, and everybody that has one letter change of this genome will have a disorder. But others, they are less deterministic, like this one in Alzheimer's disease. So let me give you the example of Alzheimer's disease that is one of the best examples of the low genetic determinism of a variant. There's a protein, a gene, that makes a protein. It's called apolipoprotein, ApoE for the short. And this ApoE comes in 3 colors. That protein comes in 3 colors. The blue, the red on the bottom, and the blue-red. And a certain fraction of the population is red. In other words, has the red allele from the father and the red allele from the mother. Homozygous in our lingo. Homozygous for red. In medical terms, homozygous ApoE4 have 12 times more risk of having Alzheimer's disease than the general population. So this is the basic of a predictive medicine based on genetic variability. So in genetics and in the genome, we can now not only diagnose people that have symptoms, as in the left part of the figure, where you see 2 affected individuals with a different color, but if we study the genome, we can identify more individuals that are at risk for this particular disorder. In genetics, we say that everybody is pre-sick, because everybody will die at some time. So that gives us a certain new view of medicine. The medicine that is based on the genomic variability on one side and the environmental variability on the other side. I'm not an environmental scientist, not an infectious disease person, but the environment is also important, as you can imagine. Or there are disorders due to the interaction between the genomic variability and the environmental variability. And we call this, the right name for this is genomic medicine. Some people call it personalized. I do not like the term because personalized medicine in many places in the world depend on the zip code where someone is born and the amount of his bank account in many countries. Francis Collins, a good friend, when he was the director of the NIH in the United States, had the ear of President Obama at the time, and he said to President Obama, let's not call it personalized medicine anymore, let's call it precision medicine. President Obama liked it. In the next State of the Union address, he said the United States promotes and supports precision medicine. Well, allow me to not to like that term either, because precision medicine we practice. We practice and practiced every day. We tried with the knowledge that we have daily, or 100 years ago, or 200 years ago, or 500 years ago, or where our great-great-great-great-grandfather Hippocrates lived, we tried to do a medicine that is based on the individual also. And Hippocrates said, idiosyncrasies, that's the right Greek word that he used, idiosyncrasia, is very important in the disorders. And we need to treat the disease, especially epilepsy, the sacred disease that he thought at the time, that is tailored to the particular individual. And this genetic variation, the medicine that is based on the genetic variation is good for prediction, as I told you, for diagnosis, of course, prognosis, and treatment, the choice of treatment and the monitoring of treatment. So in the next 5 minutes, and I'll finish, I give you some idea of what is the search to understand how the variability of the genome gives us disorders. When I started in genetics in 1979, 1980, there was one gene known at the time. That was a pre-genome, of course. One gene, the beta globin gene that causes thalassemia and sickle cell disease. That's the only one that was known. When I look at the database 2 days ago, the number of genes that we know that cause human disorders is 5,289. And we have estimated that we probably miss another 1,000 genes that cause dominant disorders. Dominant disorders need one mutation to give you a disorder in one parental allele. And we miss 7,000 recessive disorders in which one needs a bad gene that's coming from both the father and the mother. So the major search is to identify the recessive conditions that are very numerous. Now, if some of you are very interested in looking at your own DNA and you say to a medical doctor, how much you can say about my genome? How much medically you can say about genome? The answer is very little. How much? 0.43%, that very small slice of the circle that sticks out. Everything else is unknown territory for the majority of us, and your generation of scientists, you are very lucky to study all this and understand, finally, how the human genome contributes to the different disorders. In order to do this, one way to do it is to read the sequences of millions of people, millions, and compare this or associate this with the disorders that us people develop. And so far, we know of about 3.5 million of these sequences. 3.5 million. It is a very small number. We probably need, there are several estimates, but most of the estimates mean 100 million people on Earth and a very good phenotypic determination. We need to know exactly what these people develop with electronic health records and match the two big data collection and biobanks, the one in the phenotype and the one in the genotype. And the most difficult, as you can imagine, is the phenotype. Because the genotype is off the shelf. You go to a laboratory now and very easily determine the DNA sequence in a day of the entire genome. The most difficult thing is to match this with the disorders, because the disorders are difficult to extract from the people, the information, plus the phenotypes are evolving. Today I look healthy, tomorrow I develop a myocardial infarction, my same DNA. If you studied my DNA with my status now, you say you have a normal DNA. When you studied a year later, you say, oh, Some of your genes are important for your myocardial infarction. Also, our DNA is a history book. It is a history book because our humanity, when we left Africa, that scheme says 50 to 60,000 years. Put these dates with a grain of salt. Perhaps it is 100,000 to 50,000. It doesn't matter. And then we populated the earth. What people gave to the children was not wealth, but the DNA. And therefore, with the mutations that accumulate over the years, then we can say a lot about our origins, about our predisposition to disorders, and so on. I'll skip the short-term research objective of genomic medicine today for the next generations is to identify all functional genomic elements in the genome, to recognize all variants, that means type them, to say what is pathogenic. That's a very important and very difficult question. To identify all high-impact variants, because some variants give you a very severe disorder from the beginning, and other variants you need to wait 50, 60 years to develop a disorder. Devise a rapid and inexpensive ways to edit the genome. Develop computational tools for variant interpretation, to do a risk assessment, public and accessible databases, and data sharing. I repeat the words, in a fair way, and just way, sustainable way, collegial, controlled, and responsible. And most of all, as you will hear from many speakers, including my good friend Professor Gravanis, therapy. How the knowledge of the genome will help us develop intelligent and precision therapies. Then, to finish, I give you a personal satisfaction after more than 40 years in this business. And I give you an example in the gene discovery of a disorder. Some people say, so what if you identify a gene for a disorder? Well, I'll show you an example that I hope it will be put in your mind, that study the genome and understand what is causing a disorder is very important. In order to understand the recessive disorders, you need to study people that practice consanguinity. You know what consanguinity is? First cousin marriage. And why is this? Because us in the West, we don't have big families anymore and we don't practice consanguinity. But the populations that practice consanguinity, they have children with a lot of identity of the genome that the father gives and the mother gives, because they are related. Consanguinity, historically, it was practiced a long time ago. It was practiced because the power needed to be in the same clan or the same big family, or the wealth had to be in the clan and the same family. And people have understood this a long time ago. And here is a very interesting pedigree, the Ptolemy dynasty that was in Egypt from 323 before the Common Era to about 30 before the Common Era. And you see, in generation 3, Ptolemy III married his cousin, Berenice, and then generation 4, Ptolemy IV started something very unusual today, but quite common at the time. He married his sister, Arsinoe II, and had children. Generation 6, Ptolemy VI married his sister, Cleopatra II. Then an uncle-niece marriage, generation 6 and 7. Ptolemy VIII married Cleopatra III. Next generation, 8, Ptolemy IX married his sister, Cleopatra IV. And then we go down to the Cleopatra of Antonio, Liz Taylor Cleopatra in the movies, who married his brother, Ptolemy the 13th. They did not have children, and the dynasty ended there. But if you look at the map of the world today, who practices consanguinity? You see all these countries with colors, and the champion of this is Pakistan. In Pakistan, 7 out of 10 marriages are first cousins, and the population is 220 million people. So this is a very rich humanity that helps to solve the problem of variants in the genome and disorders. So we started a big project, the Swiss-Pakistani project, with collection centers in the cities that you see. I will not go into the details, but from 800 families we have identified 63 new genes for disorders, and that is unprecedented because this does not cost enormously, and in the history of gene discovery, this pace is acceleration of 100 times that was before. But I give you one example it was very important for me. This is a family that lives in the Kohat region of Pakistan. That's a very, very hot area militaristically, about 50 kilometers from the borders of Afghanistan. And there lives a family that you see here with a cousin marriage and 2 affected siblings, a male with square and a female with a circle. And you see the female, Selma, that had progressive retina degeneration, progressive blindness, and cardiomyopathy, 2 distinct phenotypes. The study of the family gave us the gene that causes that disorder, has a Kabbalistic name, SLC6A6. It's not important for you. And the mutation for the aficionados was a simple amino acid substitution, glycine to valine. Right. So then, once we know what is the gene that is important in this particular disorder, we can see what is the protein, what the protein does, what's the pathophysiology, etc. The protein is a transporter. It's a protein that takes a substance from the outside of the cell and puts it inside. And this transporter transports what? It transports taurine. Have you ever heard of taurine? Taurine is a non-essential amino acid that we say. It's an amino acid that we fabricate in our body, but we need a lot of taurine, so what we fabricate is not enough. Every time you have an energy drink, energy drinks have 1 milligram of taurine. So you're all set. You have all your taurine you need for your life. But this family cannot transport taurine in the retina cells and in the heart cells. So I will not talk to you about the mouse. That was very interesting for us because we said, since this is an amino acid substitution, the transporter did not work well, but it worked 10% of the time, a little bit of transport. We said if we give a lot of taurine to this family, to these affected individuals, who may change their lives. So we brought the family in Geneva. There was a big story, how we brought the family in Geneva. I can write a book on this. But here they are, the 2 parents, father and mother, first cousins, and the 2 affected individuals, Rifakat and Selma. Rifakat was completely blind because he was 12 years old, 13. But the little girl over there, she had some vision. She could see light, fingers, and perhaps some colors. So we decided to give them taurine. A company in Zurich made pure taurine for us, and we have a permission from the ethics committee by writing that this is not a drug, this is a nutrition supplement. So it did not take 2 years to get a permission, but only 1 month. And 2 years after the taurine, tablets of taurine from mouth, like this. The cardiomyopathy in both of them was cured. I will not give you the indices, that's for the aficionados. But they did not have any cardiomyopathy, so they're not at risk of sudden death like the football player that you've seen before. Now, the eye of the girl. This is an electroretinogram, electric signal from the retina back of the eye of her. You and I have a retinogram like the green one that you see over there. Green is normal, and red is super normal, and white is super normal. The retinogram of the girl was the blue thing that you see on your left. Blue is dead. So all the retina that she had was not functioning, except for the center. The center had a little blip of green. And 2 years after the treatment, the electroretinogram of the left eye and the right eye that you see on the bottom, they were not blue anymore. They were normalish colors. And the ophthalmologist said, several ophthalmologists saw her, she has a vision of 6 out of 20, which is for an individual that does not see, to go to 6 out of 20 is a major event, because they are independent, they live their lives, and they can have a job, they can have an education, they can read big letters, etc. So that girl was cured or improved by taking tablets of taurine. Unthinkable. Of course, that's rare, does not come every day. However, it tells you how important it is to know the disorder, know the mechanism of the disorder, and then devise an intelligent treatment based on what is wrong and not based on the symptoms. Of course, the genome could be also important for the drugs that you take today. The lady goes to the pharmacist and says, here's my DNA, give me the right medicine, etc. It will come. Now, one small problem before we finish, because nothing is bright on Earth. Sometimes it's gray, sometimes it's black. The genomic medicine has also ethical, social, legal, and financial issues. I listed them here. And I'll tell you two things: the genetic discrimination and the tyranny of knowledge. The tyranny of knowledge was elaborated and described by Sophocles, from all people. Sophocles lived from 496 to 406 BC, and he wrote this tragedy, Oedipus Rex. And in the third scene, he realizes that something is wrong with his family and his kingdom, Thebes. And then he calls the blind seer, Tiresias, say the geneticist, and he says, what's wrong with me and my town, my city? And Tiresias says, in very good Greek, Phef, phronin os dinon en thamiteli li phronundi. In other words, what do you need to know if you cannot do anything about this? In our lingo, in genetics, we call this the Tiresias complex, because diagnosis runs much faster than treatment. So we can say many things about people, but if there's no treatment, or if there's no change in lives, 50% of the people say, I don't want to know. And 50% say, I want to know, like the US elections. There are two sides equally important. And the last one is the discrimination. The reading of the genome unites people, and at the same time, it separates people. I'll give you an example from the recent history. This Dr. Robert Ritter, a very good clinician at the time, he got money from the German Association for Scientific Research to study Roma. And then in the report that he did to the funding agency, 31st of January 1941, he said, I identified, with the money that you gave me, 23,822 Roma cases that have been conclusively clarified. And you see Dr. Ritter there, examining one of his cases, subjects, and the bottom is helper, Dr. Eva Justin, does the anthropometrics in that patient, in that individual. And then we read in the minutes of the trials in Nuremberg, of the 20,943 Roma gypsies registered in Auschwitz. You see how close the number is, 20,900 to 23,800 that they registered in Auschwitz. 3,461 were transferred to other camps. You see them sitting there. They transferred to other camps because they consented to a sterilization. The rest of them died by starvation, by disease, or gas. Take-home message, proceed with caution. We need to become more intelligent, more knowledgeable of the bad history of the past, and do not make the same mistakes that we humans made before. And with this, I thank you very much, and I will say like Antonio in Shakespeare, Tempest, what is past is a prologue. Thank you.